All right, guess we'll get started. Good afternoon, everyone. I don't think George really needs an introduction to this group, but I'm sure there's some things you probably don't know about him. So I'll do a brief introduction. First thing I'll tell you is we treated him quite in style last night in dinner. It turns out that Callie's Court, the restaurant that's right down from my house, has limo pickup service for free. And the weather was so nasty that I wasn't going to let these guys walk, so we actually requested and they got a limo ride home. Anyway, so he was treated quite in style. So George actually did his bachelor's in Stockholm University in microbiology. And then he was somewhat transplanted to a very different part of the world, warmer weather, and did his PhD at the University of Crete in Greece. He tells me that part of those decisions was about which country required the most armed forces service. Both of them have some requirements, but one or the other is a little more flexible with the amount of time and what you actually have to do. He then went to Germany and did his PhD. And so by now he's picked up 3 different languages: Swedish, Greek, and German. So he's quite fluent. But he tells me in Germany they mainly spoke in English. So he was not so happy that he didn't really get to brush up on his German. He went and spent a short time in Imperial College in London doing a senior lectureship. He then came to Hopkins in 2003 as an assistant professor. In between that time and then becoming a full professor in 2011, he also did an MBA at Carey Business School. So he's got quite a wide background in terms of education. He's won several different awards, but to me, I know that this particular one, the Ellison Medical Foundation Young Investigator Award, is quite competitive. So that's a real feather in his cap. He's published a large number of publications, mainly focused on malaria, in top journals, Science, PNAS, JBC. These are all highly cited. And I think the novelty and the innovativeness of his work has mainly been to bring molecular approaches to trying to understand the processes by which mosquitoes become infected with malaria. And then understanding those, you might be able to block that infection and then the transmission to humans. So he's published many seminal papers on that process. And so it's my great pleasure to have George speak to us today. George?
Thank you, Marcia. And also thank you very much for that limo ride yesterday. I really enjoyed it. I don't get a ride in one of those Hollywood-style limos every day. Thank you, everyone, for coming here. I can't express enough what an honor it is for me to actually be considered to continue Diane's wonderful work. I work on the second deadliest animal on Earth, not because it kills itself, but it transmits human pathogens that cause disease and kill. And today, instead of focusing on one research story, I'm going to give you a little sampler of different projects that we pursue in the lab to give you a better idea of what we are about. Vector-borne diseases are complex. They involve the vector, the mosquito here in our case, the pathogen, and the human host. And most efforts to control vector-borne diseases, past and currently, have focused on interrupting these interactions between the mosquito and the human or between the human and the pathogen. My group has taken a different approach. We have focused on this interaction here between the mosquito and the pathogen to find ways to interrupt it and thereby interrupt the transmission cycle and goodbye malaria or dengue. These human pathogens, although they can be quite similar, in this case I'm showing you example of the malaria parasite and the dengue virus, have a quite similar infection cycle in the mosquito. They're ingested with an infected blood meal The female mosquito needs to feed on blood in order to produce eggs. And eventually they will infect the gut epithelium of the mosquito, where they will multiply, and they will then disseminate throughout the mosquito and end up in the salivary glands. And from the salivary glands, these pathogens can be injected into a new human host when this mosquito needs to feed on blood again. It turns out that the mosquito is a bottleneck for this infection cycle of these pathogens. And this bottleneck is found right here at the midgut epithelium. As this slide illustrates, that shows you the number of malaria parasites that you find at any given stage of the parasite's infection cycle in the human and in the mosquito. At this stage here, the parasite numbers come below 5. This is what I consider the strength of the mosquito against malaria transmission. And we decided to capitalize on this strength and look for ways by which we could make it just a little bit stronger to get that number down to 0. It turns out that that bottleneck is to a significant degree attributed to this innate immune pathway. It's activated upon pathogen recognition. It activates an intracellular signaling cascade that turn on a transcription factor, REL2, that will translocate into the nucleus and turn on expression of a number of different effector genes that produce proteins that eventually kill the parasite. And this happens in the mosquito gut. We exploited this immune pathway and this immune system to develop genetically modified mosquitoes that either overactivate or overexpress a single effector that can target the parasite, or overactivate or overexpress the transcription factor that turn on the entire spectrum of different anti-parasitic factors. So in this strategy here, we are applying something like a multi-drug therapy. You're attacking the pathogen with multiple factors. So what happens in these transgenic mosquitoes? This transgenic immune response is turned on by a blood meal because we use the blood meal inducible promoter to drive the transgenes. And the result of that is that you have an earlier immune response in the epithelium right after the mosquito has ingested the blood, as compared to a wild-type mosquito where this immune response will be mounted later on, first when the parasite is starting to invade the epithelium. And our initial hypothesis was that with this early immune response, we would actually be able to push that number down to 0. This approach has generated several genetically modified mosquito lines that are resistant to Plasmodium falciparum. You can see here the median infection level of these mosquito lines is 0. Each dot here represents the number of parasites that you find in an individual mosquito midgut. Some of you could say, okay, problem solved. We should just send a graduate student to a malaria endemic area with a big bag of mosquitoes, opening up, and goodbye malaria. We're not there yet. These mosquitoes will also have to compete and make it in nature. There's an assumption that a wild-type organism has become perfectly adapted to its environment, and any kind of engineering or tweaking you would do in its genome would do some kind of damage that would compromise its fitness. It would become less fit than the wild-type. And we decided to investigate this at laboratory conditions. So we took all our genetically modified parasite-resistant lines and we subjected them to different fitness tests. We looked at the longevity of these lines, the lifespan, how long they would live. We looked at how many eggs they would lay. We also looked at how many of these eggs would hatch. We looked at the size of these mosquitoes and how much blood they would feed. These are standard fitness tests. I'm not going to go through each one of these graphs, but what this analysis showed was that these strains did not experience any fitness loss. This was very puzzling to us. This is not what we expected, and we had a lot of trouble in publishing some of this data because of that. Only one line did experience a certain loss here, as you can see in the number of eggs it lays, and it dies off a little bit faster, but that was because the transgene had jumped into another gene and destroyed its function. The reason why we don't observe a fitness cost, we think, is because mosquitoes are a little bit like Mediterraneans. They like to take a nap after the meal, and a long nap, or a siesta. What happens after they ingest the blood meal? They go and sit somewhere and they rest for over 24 hours. They can't really fly around with their belly full of blood. So they don't really need much energy. They're not really challenged. They're sitting there, they're digesting the blood. And the blood is full of energy and nutrients. And during this rest, this is the time when this transgenic immune response takes place. It's very transient. It only lasts for a few hours. So we don't think that this additional energy required to turn on this response will have any impact on the mosquito fitness, at least at laboratory conditions. And studies in Drosophila have also shown that a transient activation of this IMD pathway does not cause a fitness cost. And strangely, these studies actually showed that the Drosophila that were transgenic and turned on the IMD pathway transiently were doing better than the wild type in some of these tests. Then we designed an experiment to look at how does these transgenic mosquitoes compare or compete with a wild type mosquito. So we reared an equal number of genetically modified mosquitoes with wild type mosquitoes in a cage as a cage population over 10 generations. And we expected to see the prevalence of the transgenics to gradually decrease and disappear, because we did think that there should be some kind of fitness cost. There should be something that compromised their ability to compete with the wild type. The result of this experiment was puzzling. Already at generation 1, the transgenic mosquitoes prevailed and established themselves at 90% in the cage population. And it got fixed at 90% throughout generation 10. And we're still having trouble understanding this phenomenon. The fact that it happens so fast suggests that it could have something to do with the mating behavior of these mosquitoes. According to a Hardy-Weinberg equilibrium, if the transgenic expression should not have any cost but no advantage, it should establish itself here at 0.75, but it jumped up at 90%. I don't want anyone to leave this room saying that the Dimopoulos group developed a mosquito that is better than a wild-type mosquito that adapted for millions of years in nature. We haven't beaten nature, I can promise you that. These experiments have been performed in highly artificial environment, in a lab insectary, where you can't really replicate all the different stresses that you find in nature. So under these conditions, we observe this phenomenon. We're now focusing on trying to understand the mechanism that is responsible for this fixation at 90%. However, the main message I want to give you here is that what these experiments show, and the previous experiments, is that this genetic engineering, this particular transgene, does not really pose a serious fitness cost to the mosquito, opening the possibility for actually using it in the field for malaria control. So the take-home message here is the IMD pathway can be engineered for resistance to Plasmodium. Transient transgene immune activation has no impact on longevity, fecundity, or feeding at laboratory insectary conditions. The transgene prevail in the mosquito laboratory cage population in absence of Plasmodium infection. A significant portion of my laboratory also studies the dengue virus interaction with its vector, Aedes aegypti. And we're obviously interested in understanding this interaction and trying to find ways by which we could actually block the transmission cycle of the virus in the mosquito gut. We have shown over the years that the TOL pathway and the JAK-STAT pathway are involved in antiviral defense, and they are blocking dengue virus infection to some degree in the gut, along with the RNA interference pathway, which is a known antiviral system. And we were interested in looking at the contribution of these immune pathways and the immune system in general in regulating resistance or susceptibility to the dengue virus in the field. So we went to different parts of the world, to dengue endemic areas in the Caribbean and South America and Southeast Asia, and collected mosquitoes from the field. We brought them back here to Johns Hopkins, and we established them as laboratory colonies. And then we infected them all in parallel with the dengue virus serotype 2. One interesting thing that you will notice is that the resistance patterns to this dengue virus serotype can vary greatly between different strains. So this Bangkok strain is almost refractory, but the Singapore strain is highly susceptible. We were surprised to see that these immune pathways seem to play different roles in regulating susceptibility, resistance to the virus in these different strains. For example, the IMD pathway appeared to be involved in conferring resistance of an Orlando strain to dengue virus, but that was not true in the Bangkok strain. And the JAK-STAT pathway, which is involved in conferring resistance or regulating resistance and susceptibility to dengue virus in the Rockefeller and the Orlando strain, did not do that in the Bangkok and Singapore strain. And in fact, in the Bangkok strain, none of these innate immune pathways appear to be involved in regulating this refractoriness. And we think that this experiment tells us that there are different bottlenecks, different mechanisms of bottleneck for the dengue virus infection cycle in these different mosquito strains. And of course, these different strains now represent a very powerful tool to identifying and characterizing resistant mechanism to dengue virus in the mosquito. Things get even more complicated if you infect these different strains with a different serotype, where now the Singapore strain isn't really that susceptible. Once the virus gets through the midgut epithelium, it will infect the salivary glands from where it can be injected into a new host. What we showed a couple of years ago was that the dengue virus will not only infect the salivary gland, it will also infect other tissues, including organs and tissues such as the antenna and the proboscis that are involved in chemosensation or olfaction, host seeking, the smell system of the mosquito. We also showed that when the antennae are infected with the dengue virus, they will change the expression of certain genes that are involved in chemoreception, opening the possibility that infection could actually manipulate some components of the mosquito host-seeking system and thereby manipulating the mosquito's behavior, possibly for its own advantage. We're not an olfaction lab. We don't work on olfaction. We don't have the know-how and the instrumentation, so we left this study right here. I know there are other labs that are more specialized in this area that are following up on these studies. Another approach of the laboratory to target the dengue virus in the mosquito is to find ways to cure mosquitoes with drugs. And I'm sure you've never heard about this kind of approach before. And we do that by targeting host factors, components of the host cell that are important for virus replication. One of these host factors are the vacuolar ATPases that are involved in the acidification of endosomes. That's important for the release of the viral RNA. Another host factor is IMPDH, which is involved in nucleotide synthesis and viral RNA replication. If you silence VATPA subunit genes or IMPDH gene, mosquitoes become more resistant to the dengue virus. If you treat any of these host factors with chemicals or drugs that inhibit them, that also results in a greater resistance to the dengue virus. We would obviously not consider using bufalomycin or mycophenolic acid as an agent, a transmission blocking agent for mosquito exposure in the field. And also this efficiency of blocking here does not appear as strong. However, these experiments serve as proof of principle that you can target dengue virus host factors in the mosquito and in that way block virus infection. And we are now searching for better compounds and better strategies to deliver double-stranded RNAs that can block dengue virus infection. The long-term goal here is to develop transmission blocking double-stranded RNAs or compounds that can be sprayed on mosquitoes like an insecticide. Instead of killing the mosquito, you would render it refractory to the pathogen. You could also expose mosquitoes to these different chemicals through artificial nectars. Because these host factors are highly conserved between the human host and the mosquito vector, some of these compounds might also turn out to be interesting for drug discovery and drug development, since they may be able to block the same host factors in the human and thereby virus infection. Take-home messages here is that the Toll, JAK-STAT, and RNAi pathways determine susceptibility to dengue virus in multiple mosquito strains, but not in all. Mosquito-resistant susceptibility patterns can vary for different dengue virus serotypes, and dengue virus host factors can be targeted and inactivated to confer resistance to the virus. I talked to you about this bottleneck earlier on, and I showed you that the mosquito's innate immune system plays a role. We have also shown that the mosquito microbiota also can influence the number of parasites that can go through this bottleneck in the midgut epithelium. And that's what I'm going to tell you about now. I will show you examples of mosquito gut microbiota that can influence its susceptibility to a pathogen in both directions. Some components of the microbiota can make mosquitoes more refractory to infection, and other components can make it more resistant, more susceptible. And in general, metagenomics and microbiota is a field that I consider very, very exciting and very relevant for public health, and it's a new field that has a lot of potential. It's also a field that ties together many different fields: bacteriology, immunology, mycology, host pathogen interactions, etc. If you dissect out the contents of a mosquito gut at around 20 hours after he has fed on a Plasmodium infected blood, you will find Plasmodium parasites in there and a large number of bacteria that can be easily isolated and grown on regular LB agar plates. If you treat mosquitoes with antibiotics in a way that you would wipe out its microflora from the gut tissue, these mosquitoes will become more susceptible to both Plasmodium infection and dengue virus infection. What these experiments are telling us is that the microbiota of the mosquito in these laboratory experiments have a detrimental role on these pathogens. They block pathogen infection to some degree. And we were interested in exploring this further, but in the field. So we went to different parts of the world, malaria and dengue endemic areas, and we collected mosquitoes. We brought them into the field lab, we dissected out their guts, and just plated out the guts on LB agar plates. We did not at this stage perform metagenomic analysis, which would give you an idea of the entire spectrum of bacteria that you could find in a gut, but we just simply grew bacteria on LB agar plates because we were interested in bacteria that can be easily cultured and thereby pursued for the development of biocontrol strategies. And through these kind of experiments, we have developed a comprehensive list of different types of bacteria, and we have brought them back to the Johns Hopkins lab, where we introduce them into mosquito together with a pathogen, and then we look at how they will influence mosquito resistance or susceptibility to pathogen infection. And we have identified a number of different bacterial isolates that can render mosquitoes more refractory to Plasmodium infection. And I'm showing you 3 examples here that we have studied in a greater depth: an Enterobacter, a Serratia, and a Chromobacterium isolate. The Enterobacter block Plasmodium in the mosquito gut. The Serratia block Plasmodium in the mosquito gut. It also shortens the mosquito's lifespan. It also produces in vitro anti-pathogen activity. The most interesting isolate is the Chromobacterium isolate from Panama. It blocks both Plasmodium and dengue virus in the respective vectors, and it shortens the lifespan of the mosquito and the larval stage. And it produces in vitro antipathogen activities. We have been interested in looking into the mechanism of pathogen inhibition that these different bacteria exert, and I will show you an example of our Enterobacter, which we isolated a few years ago in Zambia, and we showed that it blocks Plasmodium in the mosquito gut at the pre-ookinete stage. It also blocks ookinete development in vitro. So you can see here, when you have the bacteria presence, you barely find any parasites. And what our data had suggested back then was that this inhibition was to a significant degree mediated by reactive oxygen species produced by the bacterium. And we knew that the parasite is very sensitive. What our more recent data have suggested is that this is not the entire story. These bacteria also appear to produce a factor which shuts down the parasite's oxidative stress defense system, When you expose Plasmodium to Enterobacter, you can observe a downregulation of several oxidative stress defense genes, including peroxyreductase and cis-peroxyreductase. So we think that what this bacterium does, it shuts down the parasite's oxidative stress defense system, rendering it more susceptible to reactive oxygen species, both produced by the bacteria but also ROS that are present in the gut of the mosquito. And we're obviously now focusing on this factor here, and we would really like to identify this factor and characterize it in greater detail. So I've showed you examples of different bacteria that either block pathogen infection in the mosquito or kill the mosquito. How do you expose mosquitoes to this type of probiotics, if you want? Well, one way is to use their need for nectar. Mosquitoes have to feed for nectar for energy as an energy source. And these kind of nectar feeding devices, or honey traps as they're called, have already been developed to expose mosquitoes to toxins through nectars. So we are now working on looking at optimizing this kind of nectar-mediated exposure of mosquitoes to different bacteria. There's another obstacle in this approach, and that is whether a bacterium is transferred from the parent to the offspring or whether it's not transferred. So whether it's vertically transmitted or not. My bet is that most of the bacteria that we work on are not vertically transmitted. We haven't showed it exclusively, but we haven't really observed that they are vertically transmitted. What that means is that if you want to develop them into a malaria control strategy or a dengue control strategy, you would have to repeatedly expose mosquitoes to this type of bacteria. An ideal bacterium is vertically transmitted. You only have to expose the mosquito population once, and it will then propagate within the population. Automatically, it would spread through a population. And I can show you an example of such a bacterium, which I'm sure you all know. It's Wolbachia. Wolbachia is an intracellular bacterium, and if you inject it into the embryos of mosquitoes, it will establish a stable infection and be inherited from the parent to the offspring through several generations. And what we showed last year, together with, in a collaboration with one of the former lab members, who is now running his own successful lab, was that this Wolbachia infection, this stable infection in Anopheles mosquitoes, would result in a certain degree of resistance to the Plasmodium parasite. Now, this is still at preliminary stages of investigation, and of course we would have to optimize and make this effect stronger, but it looks quite promising. So now the question is, are there other interesting properties of these bacteria that could be pursued for disease control? There are. Our Chromobacterium isolate appears to produce antibiotic factors or antibacterial factors. If you streak it out on an LB agar plate, it forms a zone of inhibition that will not allow any other bacterium to grow in its vicinity. It does something more. It produces antiviral activity in its biofilm and antiparasitic activity as well, which is active against both asexual and sexual stages of Plasmodium. And here we are testing the antiviral activity of biofilms of different types of bacteria, including Chromobacterium, but only Chromobacterium appears to exert this activity. And we've obviously been very interested in identifying the nature or characterizing this type of activity. What kind of molecule is responsible for inhibiting Plasmodium or dengue virus? And we have employed a very classic natural product discovery approach, relying on a bioactivity guided fractionation, where you fractionate the biofilm of the bacteria and you test the biological activity, the inhibition ability of these different fractions, and then you further fractionate those fractions that show positive. And through this repeated fractionation bioactivity testing, we have ended up to discover an antibiotic complex that is likely to be responsible for the anti-Plasmodium activity. It's called W10. It was studied by Schering Ploeg in the 70s. And that company doesn't exist anymore. It became Merck, and Merck shut down the research facility. And I was, however, delighted to find this publication published by some investigators from Schering in the late 90s. And I thought, that's great. Now I can actually communicate with these investigators and get some more information on this W10 antibiotic. I have been trying to find these individuals. You can't see the names here. I have even considered employing a private detective to track them down. They have vanished. They have completely vanished. Okay, so we have to figure out the function and the nature of this W10 on our own. And I just want to give you, I just want to say this to the students. Think about this before you join industry. Okay? I don't know what happened. So an additional property of these bacteria is that they produce anti-pathogen activities. They can be used for natural product discovery. And I may want to remind you that many of the anti-malarial drugs today are natural product, plant-derived. So we think there is a potential in some of the components of the mosquito microbiota to also identify molecules that could serve as lead compounds for anti-pathogen drug discovery. Bacteria is not the only thing you find in mosquitoes. You also find fungi, and they have not been very well characterized in the literature. And in a field trip to Puerto Rico, we identified several different fungal species. And Ben here, he decided to actually look at whether any of these fungi could influence mosquito susceptibility to the malaria parasite. He brought them into the lab, he introduced them into the mosquito, and then he infected these mosquitoes with Plasmodium. And he got this unexpected result. When these two Penicillium species are present in the mosquito intestine, these mosquitoes become more susceptible to Plasmodium infection, significantly more susceptible by a twofold. This was very interesting. Then we asked, what happens if you introduce these fungi into our genetically modified mosquitoes that have been engineered for resistance to the parasite? So Ben performed that experiment, and something remarkable happened. The presence of these two fungi completely reverses the resistance phenotype of these genetically modified mosquitoes. And you observe a greater than an 8-fold increase in infection intensity. What this experiment suggests is that these fungi produce a molecule that in some way, or an activity that in some way interferes with the IMD pathway regulated anti-Plasmodium defense. And we don't really know exactly what is happening, but this is something that several people now in the lab are looking at. We are interested in understanding the mechanism of inhibition of Plasmodium killing. Now, you can't use this knowledge to develop a control strategy, but it is, however, quite relevant from an epidemiological perspective, and if you want to understand factors that can influence transmission in the field. These type of fungi have been found associated with different mosquito vectors, both malaria and dengue, in the past. And you can imagine how the specific composition of the mosquito microbiota in its gut can have a very big impact on transmission of disease and pathogens. So I think this is an area to focus on further. And I think it's very promising, and it will help us to understand factors that regulate transmission of disease, and maybe also to develop strategies to interfere with transmission. So the take-home message here is that a variety of mosquito midgut bacteria exert transmission-blocking activity against Plasmodium or dengue. Some mosquito midgut bacteria can shorten mosquito lifespan, and some of them appear to produce secondary metabolites of pharmacological interest. Penicillium fungi render mosquitoes more susceptible to Plasmodium infection, probably through a mechanism that inhibits the IMD pathway-regulated immune response. I've been showing you several lines of research that have a very strong translational bias. I started to work on mosquitoes in 1990, and when I started, only 13 genes had been cloned in Anopheles, Gambia. And I've had a very, very exciting journey until today, where the genomes have been sequenced and transgenic technologies have been implemented and functional genomics have been implemented. But during the most part of this journey, we were not really concerned so much about the finish line. We were more concerned about the basic science of these biological processes. But that has changed. Over the past few years, the finish line is actually coming closer. And we can actually start to see some of these technologies rolling into the field in the next maybe 5 or so years. And as we're coming closer to that finish line, we're exposed to a whole new range of matters and issues that need to be addressed, not necessarily by a biomedical scientist. There are operational, ethical, regulatory, social, political, and even economical matters that have to be resolved. And I have to admit, I'm not equipped to address these issues, but I think that expertise is quite likely to exist right here in the school. So I'm very interested, and I've already started a little bit to interact with people from other departments that are more specialists in dealing with these kind of issues, because it will become very relevant. No one has released a transgenic mosquito of this nature before in the field. However, I want to stress something here that's very important. We still need basic research. We're not there yet. We haven't reached the finish line. And we still need to do basic research. And I'm stressing this because today there is a tendency or a bias, a very strong bias towards translational research. And I'm just going to cite what an NIH program officer told me very recently. As budgets are shrinking, there is more focus on what's the practical value of publicly supported research, how to drive potential translational concepts forward. And I completely agree with this. But we should not drop basic research. Even though there is an increasing number of stakeholders today that think that we shouldn't do research anymore for the sake of knowledge only, but there should always be a product in the pipeline, further down in the pipeline. For sure, if you scrap basic research, there won't be anything to translate, and there would be nothing to apply. And I don't really know how we should get around this. Maybe we just have to become better at packaging basic research as translational. Enough with translational research. I'm going to take you back to very basic research now. I'm going to tell you about a very interesting story that has evolved in the lab over the past year or so. We have been very interested in understanding how the mosquito immune system sees and recognizes pathogens. In general, in invertebrate immunity, that is mediated through a complex of different pathogen recognition receptors that associate, they bind to the pathogen and mediate killing. So we employed an approach to identify interacting partners with one of these receptors, AGMDL1. And we used a yeast-2 hybrid screen to identify proteins that can bind to AGMDL1. In a parallel project, an independent project of this, we used an affinity capture strategy to identify proteins, mosquito proteins, that can bind to the surface of bacteria. Bacteria are pathogens of mosquitoes. So we basically mixed bacteria with a protein extract, and then we washed off everything that did not bind specifically, and then we eluted proteins that bind with high affinity to these bacteria. Both of these projects identified a mosquito cytoplasmic actin as a bacteria-binding protein and a protein that interacts with an extracellular pattern recognition receptor. This was a very unsettling result. We were not happy at all to see actin coming out here because actin is intracellular. And these two approaches were biased to identify extracellular proteins. Actin is part of our muscles. It's also part of the muscle of the cell, the cytoskeleton, and actin plays a role in a variety of processes, intracellular processes in the cell. But none of these processes have really been described as being extracellular. However, because we identified actin in two different approaches, and we verified the specificity of binding, etc., we hypothesized that, well, if actin plays a role in the mosquito immune system as an extracellular immune factor, it should be secreted by cells in an immune-inducible fashion. So when you challenge cells with an immune elicitor, they should spit out actin. So we performed a simple experiment. We took several insect immune-competent cell lines, and we challenged them with either lipopolysaccharide or peptidoglycan, two non-immune elicitors. And then we looked for the presence of actin in the supernatant of these cell lines. What we discovered was that if you challenge these cells with peptidoglycan, nothing would happen. They would not spit out actin. If you challenge them with LPS, all of these cell lines would spit out large quantities of actin. Only one cell line would apparently constitutively externalize actin. We also made sure that the cells were happy and viable when they were exposed to LPS. They're not dying, they're not lysing or anything like that. This is an active secretion mechanism. If you inject LPS into the mosquito, a few hours later, you will find actin, free actin, extracellular actin in its hemolymph. So these experiments were suggesting that actin is playing some kind of role as an extracellular immune factor. We decided to look into that, and we hypothesized that there could either be two different mechanisms it is involved in. Either phagocytosis, or it could have an antibacterial effect by directly killing the bacteria. We investigated this. We performed a phagocytic assay where we took bacteria, we mixed these bacteria with either actin or a control, and then we threw this bacteria on a cell line that was phagocytosis competent. And then we looked at the number of cells that would phagocytose bacteria. And what we observed was that exposure of bacteria to actin greatly enhanced their phagocytosis. That was also true for the AGMDL1 immune factor and when these two factors were added to the system together. And this phagocytosis was more prominent for E. coli, not so much for S. aureus, which is a gram-positive bacterium. So actin is a phagocytic factor. Then we performed an experiment where we investigated whether actin could in some way inhibit just the growth of bacteria. We took recombinant actin, mixed it with bacteria, and incubated it for a certain amount of time, either 1 hour or 24 hours, and then we plated these bacteria on LB agar to look at how many of these bacteria actually survived this treatment. And what you can see here is that there is a very strong inhibition of bacterial growth when you incubated it with actin or AGMDL or these two factors together. But that was not true for S. aureus. So there is some kind of specificity in the system. And if you look at these EM pictures here, you can see that bacteria that have been exposed to actin, they don't look very happy. And there is also this kind of mesh forming around them. If you silence cytoplasmic actin in a mosquito with RNAi, these mosquitoes become less competent in fighting bacterial infections, and they will die off faster, both with infection with E. coli or S. aureus. So this experiment shows you the physiological relevance of actin as an immune effector in the whole organism. And actin has actually been identified as immune-inducible in the past and secreted in the past. In this proteomic study from Drosophila 10 years ago, they found actin in the hemolymph of the fly after challenge, but they never paid attention to it. They never really discussed this discovery in the paper, probably because they thought it was a contamination. No one would expect actin as an extracellular factor. So the question now is, why actin? I mean, why would actin be good as an immune factor? We think the answer is resource management. The cell always has large quantities of actin in it because it's involved in so many different processes. So when it needs it for immune defense, it can just spit out some actin. It's more energy efficient to use something that you always have and you always need than having to synthesize it when you need it, or maintaining a large pool of something that you might not need. And that's what we think is happening here. And the same is true for histones, for example. They have been shown to be part of these extracellular traps, where they also mediate antibacterial activity, they enhance phagocytosis, and also they exert direct antibacterial activity. An alternative title for this part of my talk could be My Second Chance with Actin. When I was an undergraduate student in the late 80s in Stockholm, I had no intention to pursue a PhD or to do a career in research, not because I didn't like it, because I had never tried it. I didn't know what it was about. And at the last year, I had to do something which is called a diploma thesis research. I had to spend 6 months in a lab and do research. And I ended up working on actin. I was going to show that transphosphorylation of ADP actin was part of muscle contraction mechanism. And as I found this thesis last week, because I couldn't remember the title of it, I was again reminded of the fact that I couldn't get anything to work during those 6 months. It was a complete disaster. So scientifically speaking, it was a failed project. As a career step, it was a great success because I enjoyed it so much. I thought it was so fun to do science and research in the lab that I decided that was going to be my life. I was going to do a PhD and I wanted to become like my professor, Professor Lindberg. And much of this enthusiasm that I developed for science was because of my professor. And I'm mentioning this here today because I've noticed over the past few years there's a declining number of graduate students who find academic biomedical careers appealing. And when you ask them why, the answer is usually, well, the funding has become so competitive, it's become competitive and tough. And that's true. But I don't think that's the whole story. I don't think that that's the only reason. I think it's us, the supervisors, the mentors, that are failing to transmit this enthusiasm for science and to show our students what an exciting work that is. How many people get paid to work on their hobby? How many people have a job where every day is different from the other? And I think we have to do a better job on that. So the take-home message here is insect cytosolic actin is an immune factor that is secreted by insect cells upon stimulation with LPS. It binds to bacteria surface. It mediates antibacterial activity through phagocytosis and direct killing. And we think it serves a mechanism of resource management. It's more effective for the cell to use something it has all the time. Another take-home message is be persistent and patient. I had to wait for 24 years to get my second chance with actin. Some of you may ask, okay, so what's the big deal for public health with this discovery? Well, I can tell you this. If you silence actin in the mosquito with RNAi, they become somewhat more susceptible to Plasmodium infection. So we think actin is part of the killing mechanism in the midgut epithelium. And a better understanding of this killing mechanism may actually lead to the development of a new intervention strategy. So I've showed you several stories, different research lines that we pursue in the lab to find ways of blocking the interaction between the mosquito and the pathogen. And the first thing we have to do is to understand this interaction at the molecular level. I've showed you examples of different microbes that can either kill the mosquito or interfere with mosquito-pathogen interaction or produce molecules that might even be interesting for the development of drugs. I've showed you examples of how we can inhibit mosquito host factors and thereby interfere with this interaction, and also how we can genetically modify mosquitoes for resistance to the human pathogen. So I would like to thank members of my lab, current and past members that have been involved in this different research. Yesenia, Anna, Ned, Ben, Chris, Sarah, Jose, and Raul have been involved in microbiota research. April, Young, Shusmita, Nathan, Yume, Lindsay, Andy, Simone, Suzanne, Jamie, and Xiong have been involved in studying the mosquito immune system. And Narapong, or Tui, and Sekyong and Alicia have been involved in studying mosquito host factors for dengue virus infection. I would also like to thank collaborators and those that have supported these different lines of research and the different projects, and of course the funding agencies, mainly the NIH and the JHMRI, for very generous support. So I'm going to elaborate a little bit now on my vision. It's not blank. I think the department has to maintain a leading position in current areas of research and training by building on its history of excellence and by supporting, improving, and expanding its strengths. So how do you maintain a leading position? Well, first, I think you have to decide on where you want to go. I think that the department is very privileged because it has a fantastic crew, the human capital, the know-how, the competencies, the capacity, the talent. It also has a fantastic ship. The infrastructure, the core facilities, the equipment, and by being part of the school. But there's a big difference between a captain of a ship and a department chair. A captain decides on where to go, gives orders on how to navigate. A department chair facilitates, supports, and coordinates the navigation. And everyone on board should have a say. The sea is full of opportunities and challenges. There are currents and winds to take into account and to exploit to get that ship where it needs to go. So where does it need to go? Expert committees should be involved in making those decisions in the relevant fields, and they should be executing the plan, the relevant components of the plan. And yes, a plan is needed, a strategic plan, and a collective vision. So what do we need to take into account to develop that collective vision and the strategic plan? That plan has to meet current and future challenges and opportunities and needs in 4 major areas that are intimately related: public health, research, training, and funding. When it comes to public health, I think HIV, TB, malaria are going to remain the 3 big and maybe most important. But there are also many other emerging infectious diseases, vector-borne diseases, fungal infections, antibiotic resistance. Biosurveillance is going to become more important. Globalization, transport, and urbanization will pose new public health challenges. Research areas are tightly and intimately related to these public health challenges. But we also need to keep an eye on new technologies and new approaches to study biological processes. When it comes to training, it's simple. We have to attract high-quality students and fellows and provide high-quality training that prepares them for the job market. I also think we have to make academic biomedical careers more attractive. The funding trend is lesser federal funding, greater importance of philanthropic and foundation-derived funding. There is also a very strong translational bias of the funding today. So there are many, many things to consider here. But there is no department that can do everything and represent every possible area or discipline. And you really need critical mass in any given area to make an impact. So what I think is a reasonable strategy is to focus on current strengths and nurture, enhance, and expand them. So what are the current strengths? First, it's the human capital, the talent, the capabilities, the expertise, and the know-how. And the department is making a very significant impact today in immunology, parasitology, bacteriology, vector biology, virology, disease ecology. It's quite broad. And that human capital has to be nurtured, supported, but also strengthened and expanded with new talent that will complement existing expertise and be integrated in the existing intellectual community. New talent that will safeguard the department's route into the future. The demographics of the department, the need for new talent, and the availability of candidates will determine the direction of these expansions. But expansion is not everything. I want to make that clear. I think it's extremely important to support the current capacity. When it comes to infrastructure, I think the department is pretty top-notch, especially if you look at it in the broader Johns Hopkins community. It has an excellent training program. But expert committees will have to make decisions on the expansions and the changes that have to take place in order to meet the challenges of the future. I find metagenomics and microbiomes a very promising field. Systems biology, pathogenesis, field and disease ecology, human immunology, different omics-based approaches to do research, and also medical entomology. I know there is a desire to upgrade some of the infrastructure, BSL-3 and gnotobiotic facilities, but I also think we need to consider the Johns Hopkins wide complementarity when it comes to facilities. We don't want to duplicate. It's not cost effective. When it comes to training, I think we need to look at whether we have unutilized know-how that could be translated into new course offerings. We also have to look at the comprehensiveness of the training program. Is it too comprehensive, Could it be redesigned? Is it up to date? I think we need to put a greater emphasis on career management training for our students. In these volatile times, things can change very rapidly. New public health challenges and needs can arise. New technologies arise. And the funding environment and opportunities changes continuously. So agility is needed to meet these new challenges. And the strategic planning process is not something that only happens once and then we just follow the plan blindly, but it's a continuous process. It happens continuously. You scan the internal and external environment for challenges and opportunities, and you have to make corrections to the course in order to meet your objectives. I can't speak as an expert for virology or bacteriology or even human immunology. I can't do that. Expert committees will have to do that. But I can speak for medical entomology, which I think is a very promising area, in particular for the MMI department. First, it has a huge public health relevance, especially when it comes to emerging infectious diseases. And I'm not referring to malaria here. I'm speaking about dengue, chikungunya, West Nile virus. When it comes to research, it's one of those disciplines that tie many disciplines together. Immunology, host pathogen interactions, virology, bacteriology, metagenomics. I've highlighted these 3 nodes here because they represent unique assets of the department. The human capital, we have 5 groups that work on medical entomology today. Research infrastructure is really state of the art. We have insectariums and parasitology core facilities, field facilities that you don't find elsewhere. And there's an excellent training track record and course curriculum. And if you look at other immunology and microbiology department, I'm mainly speaking about those that you find in the major Ivy League universities that we compete with for students. This is something that we have that no one else has. And many of our students, they come here because of medical entomology. So this is a field that I believe in. And I think there is a lot of room for expansion there. But again, I can't speak as an expert for the other fields. And I'm not want to give you the impression that I have an exclusive interest in this field, and I think this field should be exclusively enhanced. But it's a field I can speak for. Thank you, and I can take questions. Yes.
So, your first example with the IMD pathway, are there naturally occurring alleles that have of the genes in that pathway that have different levels of activity?
So, of the pathway itself, these pathways are highly conserved, the intracellular part. So, I'm not aware of allele polymorphisms that could confer different levels of resistance, but some of the effectors that are controlled by the IMD pathway. Some of them have been studied, and there are some alleles that confer different levels of resistance in the field. Yes, Marie?
I was wondering about how much actin is being released, and for example, would it compare to, say, the lysis of 1 cell in a million, or more, or less?
It would definitely, well, I actually can't answer that question. It's not something we have looked at. We have done different type of cell viability assays, both chemical and microscopy, and we cannot see any cell lysis whatsoever in these experiments.
I guess if it's lysed.
Yeah. Yeah, well, I mean, usually you see cell debris. If something lyses, it leaves debris behind it. And there is a lot of actin that is spit out in that supernatant that I don't think, it's a lot, yeah, it's a lot.
Does that correlate then with the amount required to inhibit the bacterial growth?
Oh, yes, so the experiment which we designed, the phagocytic assays and the antibacterial assays, we actually paid attention to correlate those amounts of actin applied to the physiological levels of actin that you find in the supernatant. Well, oh yes, so we did find other proteins, a variety of other different factors, and we were looking for things that maybe could be interacting with actin, and there's nothing clear. We also find, immune factors and things that have been discovered in the past. But we found actin particularly interesting because it has never been studied in this context before. Yes, Clive.
How consistent are these infection, these different viruses, these different bacteria that you find in fungi in populations of mosquitoes from the field? Or are they dynamic or are they opportunistic?
Well, that is a question that I would really love to pursue as a research project. We haven't really had the opportunity to do surveys in the field of that breadth that would answer that question. And it's very difficult. There are only 2 different metagenomics-based surveys of mosquito microbiota that have employed field mosquitoes, so you can't really use the data that they generated and generalize from there. But you do find Enterobacter as a very common bacterium in different Anopheles species, for example.
Because actually that becomes important if you try to translate that.
Yeah, and that would be something that we're very interested in doing, to basically correlate mosquito infections with either Plasmodium or dengue with the microbiota to identify correlations that would suggest that certain types of bacteria would inhibit the infection with either one of these 2 pathogens. Yes?
I have a couple of questions. The first one is, do the mosquitoes have more than one microbiome? So is it just the gut or is there something else? And the second one, if you want to answer that first, maybe.
Okay, do the mosquito have different microbiomes? Well, the gut has been studied, but I also know that the Malpighian tubules, which is the sort of kidney analog of mosquitoes, is also full of bacteria. And I think there are some reports that have also showed that the ovaries may carry some bacteria. I'm not aware of the salivary glands. Well, there is actually one bacterium, an Asaya isolate, that has been found to be associated with the salivary glands of the mosquito. But those other microbiomes than the gut microbiome, they have not been really extensively studied.
Okay, and as a follow-up question, kind of piggybacking off Clive, is can you use computer modeling perhaps to look at all the microbiota in certain mosquitoes, and then you can maybe see a pattern of various combinations of bacteria that would work best?
Yes, absolutely. We would have to do the metagenomics analysis first to get a picture of the entire spectrum of bacteria that you find in samples from the field infected with a human pathogen or non-infected. That's what we would like to do, to identify patterns that suggest that certain bacteria are either promoting or inhibiting infection with these human pathogens. Yes?
George,
Oh.
with the actin response, you mentioned, I think, that it was also observed in Drosophila.
Yes.
What about mammalian cells?
Well, there is one study that has showed that HIV-infected cells, I can't remember exactly which cell type, results in the externalization of actin, but at the very surface of the cell. Yeah.
So as you pointed out in your genetically modified variant of strategy, and you also showed that the 2 penicillin strains reconstitutes susceptibility. Now, if we go into the wild, do you think that even if you release a genetically modified mosquito, would that hamper your efforts?
Well, yeah, possibly it could. We actually don't know the prevalence of these particular fungi that have this type of activity in nature, A. B, we actually don't know yet, but it's something we're looking at right now, whether the infection intensity with a fungus that we can achieve in the laboratory reflects what you find in nature. So we feed the mosquitoes on a nectar solution that contains fungi. And we may actually be exposing the mosquitoes to a very large, unnaturally large quantity of fungi. We don't know that yet. But it is possible that it could interfere with these type of strategies. Yes.
Beautiful talk.
Thank you.
I'm just curious, you alluded to the idea of thinking about the safety implications of releasing a super mosquito.
Yes.
How would you think about that?
Yes. Well, You know, we have actually, that's something that we did think about at the quite early stage of our experiments, and I didn't have time to elaborate on that, but you know, these genetically modified mosquitoes, they don't express a foreign gene because we used their own immune activator gene, REL2, fused it to its own promoter, a promoter of another gene that is turned on by a blood meal. So we just changed the expression pattern of its own gene. So we don't express anything foreign in that mosquito. So we haven't changed it dramatically. The only thing we've done is we've changed the activation pattern of its own immune response. We are looking right now at whether this mosquito has become more or less susceptible to other human pathogens, such as Onyong virus that is transmitted by Anopheles mosquitoes. But I can't really see any other safety concerns, if you want. I think the worst possible outcome would be that the parasite would develop resistance to this mechanism over time. But there will be concerns. Certainly there will be concerns, and that is what needs to be addressed. Yes?
Could you perform further genetic modification to your anti-Plasmodium transgene mosquitoes so they could also be resistant to penicillin?
Possibly. I don't know how many transgenes you can put into the genome and still having a happy and strong mosquito. Yes?
Following up on the actin, what's your speculation about how actin is activating phagocytosis? Does it activate phagocytic cells in terms of cytokine production?
Yeah, so the insect immune system is a little bit different from the mammalian. So there are no same type of cytokines that you find in the mammalian system. We haven't looked at that. We haven't actually exposed host cells or insect phagocytosing cells to actin yet to look at changes. We are now looking at the action of either F-actin, filamentous actin, that polymerize and form long filaments, versus G-actin, and how that may differentially be involved in these different mechanisms. Our preliminary data suggests that F-actin is also mediating, and both F and G-actin are mediating the phagocytic process, but F-actin does not seem to have antibacterial activity. What I think is happening is that actin is in some way inserting itself into the structure of the cell wall and causing some kind of inhibition because it binds to it at a very high affinity. Even after several washes, high stringency washes, we can still find actin associated with this bacteria when we just run the entire bacterium, whatever is left after the washes, on a Western blot.
On the surface?
On the surface, yeah. But we don't know the mechanism. I mean, we speculate.
Do you know if the actin has any post-translational modifications that's associated with this yet, or do you suspect?
We don't know. I mean, we produce recombinant actin in a yeast in an insect cell line system, I believe. But it's something very interesting to look at in the mammalian system, which we haven't done.
Thank you.
Thank you.